So welcome everyone. This is our 4th webinar of our series. And this one is one of the webinars that's focused on Ewingssarcoma. It forms part of our work that we're looking at in Ewingssarcoma. So before we start with the webinar, I just thought I'd go through some of the kind of housekeeping bits. So if could everyone keep their microphones on mute while our experts are talking, so that they don't get distracted and they can give their presentations and you can all hear nicely. If you've got any questions, we do have a comments box. So if you write your questions in there, Vicky, my colleague, will collate them all and there's a chance at the end for those to be answered. We have had some questions in advance and we'll go through those as well and our experts will answer them. So the webinar is very much research focused, laboratory research. If there's anyone that's got questions about their current situation, their treatment, or any kind of clinical questions, we might not be able to answer those in the webinar itself, but we will get back to those people who have the questions outside of the webinar. So today we're joined by 2 speakers. So as part of our 2017 to 22 strategy, we wanted to make sure that BCRT could support researchers at every stage of their career, from kind of students up to principal investigators. And we wanted to pay particular attention to developing the next generation of researchers that could take forward primary bone cancer research. And so it's really, really nice to introduce our speakers today. So Tyler is a PhD student at the University of Leeds, and she is joined by her supervisor, Dr. Fiona Errington-Mais, who is a lecturer at University of Leeds too. So I'll hand over to them and they're going to give you an update on immunotherapies and some of Tyler's work. So I'll just share my screen. Just bear with me. Just bear with me. There we go. This should be sharing now. Can you all see that? Is that sharing? Try again.
We could see it, Zoe. All right.
Oh, could you?
Yeah, yeah.
Yeah, is that good?
I can see it. I haven't got the proper the slideshow presentation. It's still in editing mode.
Yeah.
It won't click onto the. Is that better?
If you look at the top, Zoe, it's where it says enable editing. Can you click on that? Do you see that?
I think you can just press F5.
Oh, she's going to cut everyone off.
Yeah. That not work?
No.
It won't change.
Yeah, I think you need to do the enable editing.
Unshare it, put it onto slideshow, and then reshare it. Does that make sense?
Oh.
Hurrah!
Yes! There we go. Over to Fiona.
Oh, I just had a major panic there as well, because the whole screen just disappeared for me and I couldn't see anything. So I was like, this isn't going to be good. Okay, so thank you to the organisers for this, for giving us the opportunity to contribute to this webinar series, and particularly to the previous speakers who have given excellent overviews of how bone cancers develop and some of the challenges associated with the treatment of those bone cancers, and to Ali last week, who talked us through the dynamic organ of the bone. So today, Tyler and I are going to do a double act, and we're going to really talk to you about some of the work we do, which is aimed at manipulating the immune system to try and develop a novel treatment for Ewingssarcoma. Next slide, please, Zoe. So as you can see, I'm working from home. This isn't my bedroom, by the way. I've kicked my daughter out today to be able to get on and have a nicer working space for you all. But as you can see, I am at home, and there may be connection problems like there were last week. So if anything goes wrong and I start talking to myself, please start waving to let me know that you can't hear me. And if I can't see that, someone's going to contact me, but at least you'll keep each other entertained by waving at each other as we go through. And I also have 2 kids and a dog at home who pick the worst moments to come and see me and have arguments and fights. So I apologise if they interrupt you at any stage during the presentation today. Next slide, please, Zoe. Can everyone hear me okay? Because I think Matt said before I was a bit muffled. Is it okay, Tyler? Yeah, okay. So the immune system, today I'm going to talk to you about the immune system and really how its role in the development of cancer. And in particular, I'm going to focus in on some of what we know about the immune system in the development of Ewingssarcoma. After that, I'm going to talk to you about some examples of immunotherapy, but with a particular focus on our area of expertise, which is oncolytic viruses. So I'm going to talk to you about how they work, but also show you a clinical example of how they've been used in patients to hopefully convince you all that they can work as a cancer therapy. And then Tyler's going to come in for the second part and talk to you about the exciting bit, which is funded by the BCRT. Next slide, please, Zoe. Thank you. So our immune system is made up of many different cells and molecules, such as cytokines and chemokines, that are all tightly regulated within the blood, the bone marrow, and throughout the body. And why are these important? So their everyday job is to fight virus and bacterial infections, recognize those infections, eliminate them, and make us all feel better. But how is that relevant to cancer? So as Darryl talked about a few weeks ago, cancer is made up of, develops because of genetic mutations, which causes the development of unusual and non-self proteins within the body. And these non-self proteins can be recognized by T cells and NK cells for elimination within the body. And there's other immune cells, such as macrophages, which can play different roles within the tumour microenvironment. They can either have pro-tumour properties or anti-tumour properties. Next slide, please, Zoe. So within Ewingssarcoma, they get the chromosomal translocation. And what happens as the cancers develop is that you tend to have this elimination phase, where the cancer cells are recognised as foreign. They can be eliminated by T cells and NK cells at this early stage. But what happens is you end up with this kind of homeostasis, where you have your cancer cells trying to modify themselves and maintain existence, but you have the immune cells trying to fight them off. Eventually, unfortunately, the cancer cells win, and you end up with this really complex tumour microenvironment, which switches off all of the immune cells within the effector immune cells that have the anti-cancer properties within the tumour microenvironment. And you also get the recruitment of other immune cells which aid the switching off of those effector cells, and they can also help promote disease progression and cause metastasis. Next slide, please. Sorry. So what do we know about this in the context of Ewingssarcoma? Well, I think it's safe to say that actually in the context of primary disease, we're not sure how this actually works. But certainly in the context of metastatic disease, there's been a lot of these immune evasion strategies that have been identified. For example, you can get a high number of regulatory T cells or myeloid-derived suppressor cells within the tumour microenvironment. And these actually secrete lots of regulatory cytokines which switch off to NK cell and T cell function. We have the Ewingssarcoma cells which have adapted to hide away from the immune cells, so they can downregulate things like MHC class 1, which is a molecule that's recognized by T cells, but they can upregulate these immune checkpoint molecules which make them unable to interact with the T cells and switch those off. So basically the Ewingssarcoma cells hide from the immune effector cells and switch off the immune cells that they engage with. Then you have the high levels of these M2 macrophages, which secrete cytokines which enable the tumour to grow, and as Darryl said, might play an important role in metastases. And then you have this really rich microenvironment, which is full of TGF-beta, which is a cytokine that really switches off your NK cells and T cell function. So you have this really complex environment with multiple challenges that you need to overcome for any immunotherapies to work. Next slide, please. Sorry. So really we have this battle between the immune cells and the cancer cells. So initially the immune cells win and eliminate the cancer, but as the cancer develops, you have a lot of cancer-driven immunosuppression mechanisms, and the cancer wins, unfortunately. Next slide, please, sorry. So what immunotherapies are thought to try, or are designed to do, is really wake up the immune system to be able to make it capable of killing and recognizing those cancer cells. So you can either do that within the cancer patient, or you can deliver immune cells capable of killing the cancer cells by infusion into the bloodstream. So hopefully by using these immunotherapies, you can regain immune control of the cancer. Next slide, please. So this is some examples of immunotherapies, and I'm by no means an expert in any of them, apart from probably this one. I would classify that this is my area of expertise, the oncolytic viruses. But just to give you an overview of the types of strategies that exist. So looking here at the monoclonal antibody therapies, there's molecules out there that are known as immune checkpoint regulators. So we know cancer cells can regulate things that turn off your immune effector cells. One of those in Ewingssarcoma is PD-L1. So what people have done is develop antibodies that block that inhibitory interaction and maintain the function of the immune cells and enable it to kill. So that's one monoclonal antibody therapy. Another type of monoclonal antibody therapy is really just aimed at bringing the cancer cells and your immune cells into close contact with each other. So essentially the antibody will recognize a target molecule on your cancer cell. The Fc portion of the antibody will interact with the immune cells and bring them together and allow the immune cells to kill. Typically that's a function carried out by natural killer cells. Moving on, you can have the adoptive transfer of immune cells, and an exciting therapy at the moment are these CAR T cells. So these are genetically engineered T cells that are engineered to specifically recognize molecules on the surface of your cancer cell, again bringing them into close contact with each other and enabling killing. Or you can transfer in activated NK cells that have been bulked up in the laboratory and then infused back into patients. At the right-hand side, we have this kind of global cancer vaccine, and this incorporates lots of different types of therapy. But a vaccine is really based on how a flu-like vaccine would work, essentially where you inject bits of the molecule, the virus or the cancer cell, into the patient to try and develop a host immune response towards that molecule. And cancer vaccines can be carried out either with modified cancer cells that they put into patients. you can use dendritic cells, which are a classical antigen presenting cell, as we call them. So they present bits of the cancer cell on their surface in an appropriate manner to help boost T cell responses. Or you can just put in bits of peptides and proteins from the cancer cell to try and facilitate a T cell immune response. And then you come to what I think is obviously the most exciting immunotherapy, which are your oncolytic viruses, which are cancer bursting viruses. And the reason I've put these little arrows encompassing these other strategies is because actually oncolytic viruses can engage and act like many of these other immunotherapies. So they can work in synergy with your monoclonal antibody therapies to enhance therapy. They activate T cells and NK cells to kill. And really what they are is an in situ cancer vaccine. So as they are killing your cancer cells within the patient, they can release tumour antigens and prime up these immune responses towards the cancer. Next slide, please, Zoe. So how do oncolytic viruses work? They're considered really to have a dual mode of action, although lots of other mechanisms are coming to the forefront now. So oncolytic viruses preferentially replicate within your cancer cells and leave healthy cells unharmed. And this is really important because what that means is you get very little off-target effects with these viruses. As you would with chemotherapy, they kill lots of non-cancerous cells. Viruses tend to specifically replicate in cancer cells and cause this lysis. But also, because they're a virus, they're seen as foreign, so they're very capable of activating various arms of the immune system to also kill the cancer cells. So you get lysis of your tumour cell and you generate this inflammatory cytokine-rich environment. Many of these cytokines can actually have direct killing effects on the cancer cell. The cytokines released also activate the NK cells within the tumour microenvironment to kill the tumour cells, but also dendritic cells within the environment can take up bits of these tumour cells, present them to T cells and prime it and help generate cancer-specific cytotoxic T cells. Next slide, please, Zoe. So just to prove to you really that oncolytic viruses can work, and they've been tested in numerous clinical trials, and there's a range of oncolytic viruses available out there to clinical grade, which is excellent. Overall, every trial that I've seen, they've been extremely safe and very well tolerated, and usually a lot of the symptoms can be controlled with things like paracetamol because they just tend to generate a fever associated with a virus infection. And there is evidence to date that shows that they can work in combination therapies with other agents, but also as single agent therapies. And the most clinically advanced oncolytic virus is Telamogene laherparec, or TBEC, which is what I'm going to say on the next slide because it's too much of a mouthful. And this is the first virus to be FDA approved for the treatment of advanced melanoma. Next slide, please, Zoe. So this is where I aim to get you all extremely enthusiastic about this therapy. So this is just some data for the phase 3 controlled clinical trial with the TBEC agent. Like I said, it was used to treat extremely advanced melanoma that was unresectable. And immunotherapies tend to take quite a long time to work. So what they did in this trial was they injected every surface melanoma lesion that they could access every 2 weeks for up to a year. And they injected up to 4 mils of virus across multiple lesions in each of the patients. The virus was extremely well tolerated in those patients, and it caused regression over time, possibly by about 6 months is when you saw most of the regression really starting to come to its forefront. But you saw regression of the primary lesions, but importantly, I think for the context of Ewingssarcoma, is you also got regression of distant metastases in the lung, which is excellent for Ewingssarcoma because that's a common site of metastases for that disease. But you also saw regression of the disease in the liver, peritoneal, and other non-injected lesions. And the reason that is extremely good for us is that actually that really demonstrates that you're generating this systemic immune response to the virus after you've given it into the tumour site. And this is just to show the survival curve data for that trial, with the blue line being the virus treatment on the top and the yellow line being the control arm at the bottom. And as you can see here, you got an increase in overall survival in the patients that were treated with the TVEC. Next slide, please, Zoe. So overall, what we anticipate for this virus is that actually we think standard of care is really good at treating the primary disease. And actually we know a lot of those immune evasion strategies are probably more prevalent in the metastatic setting. So what we would propose to do is use standard of care to treat the primary disease, allow the immune system to regain function, because obviously we know chemotherapy can cause lymphopenia and cause your immune cells to become a little bit sick. And then you would go in with your virus infusion, plus or minus any complementary agents that might enhance its efficacy, to hopefully then eliminate any residual cancer cells that remain in the body, either via direct lysis or activation of the immune system, and then hopefully the disease will not relapse going forward. So now it's over to Tyler, who's going to talk to you about some of the exciting data that she's been generating in the lab relating to the viral therapy work we've been doing. You're on mute, Tyler.
Yeah, I'm here now. So hi everyone, I'm Tyler, and today, oh, the slides have gone. Vicky, sorry Zoe, would it be easier if I just shared the slides?
I found it a bit, I was pointing at things and I thought that people could see what I was pointing at, but I don't think people could actually, could they?
Oh, there we go. Right, okay. So today I'm presenting about our research project. So the aim of our research project was to provide preclinical data, basically which just supports evidence for Ewingssarcoma treatment with oncolytic viruses, and so that this can evidence to see if this can be translated into clinical trials. So I've just set out here multiple steps in the research project. So as Fiona described, there is a dual mechanism of action of oncolytic viruses. So to investigate this first mechanism, I wanted to determine if the oncolytic viruses could kill Ewing-sarcoma cells directly. Also, I wanted to assess the ability of the viruses to kill cell lines which are resistant to chemotherapy. So it is quite well known that chemotherapy resistance can be a big challenge in Ewing-sarcoma. So this was quite important to determine if any new therapies that are developed are able to kill cell lines that are also resistant to standard of care treatments. So the third point, assessing the second arm of oncolytic virus mechanism. So we wanted to assess the ability of the viruses to activate immune killer cells or natural killer cells against Ewing-sarcoma cells. And then point 4, we wanted to combine the oncolytic viruses with other drugs, particularly antibodies we're quite interested in, so other immunotherapies. And point 5, in the future we want to use patient samples. At the moment we use patient cell lines, so these are grown in the lab over many years, so patient samples more accurately represent the disease in the lab. So next slide please, Zoe. So on to that first point, testing the ability of oncolytic viruses to kill Ewing-sarcoma cells directly. So we have a number of Ewing-sarcoma cell lines, and these are cell lines are derived from patients, but they've been grown for many, many years in the lab. So the cell lines that we use are SKNMC, SCES1, TC32 and TTC466. I just thought I'd put these here to start with so that when they pop up in all my slides, you know that they're the Ewing-sarcoma cells. So we also have a number of oncolytic viruses which we're looking at. So there's reovirus, a rhabdovirus, herpes simplex virus and an enterovirus. So what we do to test these in the lab is we treat the Ewing-sarcoma cell lines with the viruses for 48 hours. Other time points have also been looked at. And then we add a yellow fluorescent stain to the cells after treatment. And the dead cells take up more of this yellow fluorescent stain. And then using this really, well, my favourite technique called flow cytometry, we can then look at these cells. If you look at the little plot in the bottom right-hand corner, and the cells which are on the bottom axis, they basically take up more of the live dead stain. You know that they're the dead cells. So after treatment, you can effectively look at how many cells within that population the virus has managed to kill, and therefore it tells you how effective that virus is against Ewing-sarcoma. Next slide, please, Zoe. So the results for that are here. So for reovirus and herpes simplex virus, you can see a dose response. So as you increase the amount of virus you put on, you see more cell death. And the maximum we really see is around 50% cell death with those. With the rhabdovirus, you can see quite high cell death. And even at 0.01 PFU per cell treatment of virus, you see up to 80% Ewingssarcoma cell death. So yeah, that one looks the most promising in terms of direct killing effects. The enterovirus, unfortunately, it shows that the Ewingssarcoma cells are quite resistant to that virus. So we actually just took the other 3 forward for further investigation. Next slide, please. So on to the next point, as I mentioned earlier, it's important to look at chemotherapy-resistant cell lines. So Ewingssarcoma tumours can become resistant to standard of care chemotherapies, as can a lot of different types of cancer. So it's important that any new therapies that you investigate are able to kill not only the primary tumour, but also any relapsed and resistant to therapy tumours. So we have this cell line which is resistant to doxorubicin, and this was generated by Professor Sue Birchall and Dr. Elizabeth Roundhill at the University of Leeds. And basically what they do is they get this cell line called SKNMC and they treat it with doxorubicin at low doses for a prolonged period of time. And this makes it resistant to doxorubicin. So you can see from the figure on the left, the green line shows the parent cell line, just the normal non-resistant. And then the orange line shows the doxorubicin resistant cell line. And you can see from both of those lines that you require more doxorubicin to kill the resistant cell line. So this is just to show you all that is in fact resistant. So on the right, you can see that we've challenged both of these cell lines, the SKNMC and the doxorubicin resistant cell line, with the rhabdovirus. And you can see that both cell lines, that rhabdovirus is able to kill both of these cell lines, and despite it being doxorubicin resistant. Next slide, please, Zoe. So moving on to the second arm of oncolytic viruses, and there is immunotherapy, so their ability to activate immune cells. So we're particularly interested in these immune killer cells, or natural killer cells. So we can obtain these from blood samples, and we treat them with the viruses. And you can, when the natural killer cells are activated, they express these molecules called CD69 and CD317. So what we do is we obtain these immune cells and treat them with different doses of virus, or as a control with no virus. And then you can look at the changes in these markers, CD69 and CD317, and it basically tells you that the oncolytic viruses are able to activate the immune cells. Next slide, please. So you can see the results here for reovirus, rhabdovirus, and herpes simplex virus. And you can see that there's a nice dose response. So if you increase the amount of virus, you get nice activation or increased expression of those CD69 and CD317 activation markers. And actually you can see for the rhabdovirus, you can, after rhabdovirus treatment, you get up to a 20-fold increase in the CD317 marker. Next slide, please. So obviously we've shown that the oncolytic viruses are able to activate these killer immune cells, but it's important that once activated, they're actually able to kill Ewingssarcoma cells. So I've created a little video, and here what you'll see is that there's killer immune cells which have been pre-activated with oncolytic rhabdovirus. They are then co-cultured in a dish with Ewingssarcoma cells, and the Ewingssarcoma cells have been stained green. So any other cells that aren't green, they're the immune cells, the killer immune cells, and the green cell is the Ewingssarcoma cell. And in the bottom right-hand corner, you'll see the time, which is in minutes. So you'll see over a very short amount of time, the immune cells are able to attack the Ewingssarcoma cell after virus treatment. So the video is playing now. So you'll just see there that the immune cells are very quickly attacking that Ewingssarcoma cell. And that's just in a matter of 40 minutes. So yeah, if you could, just play that again. There we go. Yeah, I just thought I'd add that in because I can show you a lot of different figures and bar charts and things like that, but it's good to see it just happening sometimes. So yeah, just next slide, please, Zoe. So obviously we're all very thrilled by the natural killer cells. This is because there's a lot of interest in natural killer cells in terms of oncolytic viruses and in Ewingssarcoma as well, prior to all of the oncolytic virus work. And this is also one of my supervisor's favourite cell types. I won't tell you which one. Yes, so we needed to make sure that the natural killer cells, are they in fact the most important immune cell in terms of oncolytic virus therapy for Ewingssarcoma? So I set up an experiment to determine this. So I have Ewingssarcoma cells, which are stained with a green fluorescent stain. And then alongside this, I have some immune cells, which I've just derived from blood. So these are just all the immune, like white blood cells that we have. And then I also have these immune cells, but I've depleted them of the immune killer cells. So I treat these two populations of immune cells with oncolytic viruses. Then bringing all of this together, I co-culture either the complete immune cells or the immune cells depleted of the immune killer cells with the Ewingssarcoma cells to assess their ability to kill the Ewingssarcoma cells. So if you go into the next slide, thank you. So first of all, this result just shows you that the whole immune cells were treated with either no virus as a control or each of the three viruses. And then when they're co-cultured with the Ewingssarcoma cells, this is showing you the percentage of cells that are able to kill. So as a control with no virus treatment, immune cells can kill around 40% of Ewingssarcoma cells. But then after you add virus, this increases to 70 to 80%. But this is the interesting part, so if you could just move on to the next bit, Zoe. When you deplete the immune killer cells, this effect is significantly reduced. So you don't see this enhancement of their ability to kill the Ewingssarcoma cells, which tells us that it is in fact the immune killer cells which are responsible for killing the Ewingssarcoma cells after virus activation. So knowing this, we can investigate combination therapies because there are antibodies available to enhance immune killer cell mediated attack. Next slide, please. So going back to this diagram which Fiona showed you, which is basically the balance of immune cell activation and cancer-driven immune suppression. And oncolytic viruses work very well in the lab, particularly at activating immune cells. But when taken to clinical trials, sometimes they can not work as well as you've seen in the lab. And this is thought to be a result of the immunosuppressive effects of the tumour microenvironment, which Fiona described. So what you need to do is you need to add in other agents to help tip this balance. So we are investigating combination immunotherapy of oncolytic viruses with antibodies. And these antibodies have already been developed clinically, and they can block the inhibition of immune killer cells. Next slide, please. So killer cells are controlled by immune checkpoints. So I'm sure everyone has probably heard of these at some point, as they're on the news quite a lot, as immune checkpoints have been quite important in T cells. So in a similar way, immune checkpoints on NK cells basically act as regulatory molecules. And NK cells have activatory molecules which tell them to go and attack, but they also have inhibitory molecules which send a stop signal. And this is to make sure that obviously they don't just kill all the healthy cells in your body, for example. So effectively, what we want to do is we want to block this inhibitory signal that the cancer is expressing a ligand to this inhibitory molecule, and then we want to try and block the inhibitory molecule. So lirilumab is an example of an antibody that has been developed, and it can block the inhibitory molecule on killer cells. So we are now working on looking at combining these agents with oncolytic viruses in the lab. And as I said, it's just to try and tip that scale for immune activation as opposed to tumour-induced immune suppression. Next slide, please. So just the final point that we're working on, we plan to repeat all of these experiments that I've told you about and all of the other experiments that I haven't had time to tell you about using patient samples. These are much more representative of patient tumours, and it would just give us, we just need to ensure that these agents all work in the patient samples too. I haven't touched on this as much today as I have mentioned this before in lab visits, so I don't want to keep talking about the same thing. But we're also working on developing a more representative Ewing-sarcoma model in the lab. So Fiona expressed to you the importance of immune cells in the tumour microenvironment, but there are also other cells that come into play, particularly in Ewing-sarcoma. So there have been a lot of publications about osteoclasts, which are our bone-eating cells, and fibroblasts, which form the tumour stroma, so the area around the tumour. And there have been lots of reports of these cells contributing toward tumour invasiveness and driving the tumour to grow. So we want to make sure that these won't hinder oncolytic virus therapy, and we can also look to see if the viruses are able to impact these cell types and are they able to kill the tumour-associated fibroblasts. Next slide, please. So in summary, our results so far are quite promising for oncolytic viruses for the treatment of Ewing-sarcoma. The direct killing effects of the virus, I've just summarised that here. For reovirus and herpes simplex virus, they look quite good, but much better for the rhabdovirus. The enterovirus wasn't so promising, so we didn't take that forward. The ability of each of the other viruses to activate immune cells against Ewing-sarcoma are quite equal. So for this reason, we are taking all of those other three viruses forward for further investigation. It's very important for us to emphasise, especially as we are all, myself and my supervisors are all researchers, we're not clinicians. So this is very preclinical project, this is all lab-based research, and translation of these results into clinical trials can be a lengthy process. But the important thing that I want to emphasise is that without this preclinical evidence, the clinical trials would not be possible at all, and that is why these projects that BCRT support are so important. Thank you. So finally, I just want to say a very big thank you to everyone for your support at BCRT and all the supporters. I just put this little picture in here. I lost a friend in 2015 to Ewing-sarcoma, and his lovely mum Bev has been making these bears to raise money for BCRT, and it's just a lovely example of all the things, the imaginative things that you all come up with to try and support the charity. Thank you to everyone in our institute at our university. They've helped us with bake sales, and we did plan to all do a dragon boat race again this year from our university. Hope we can do that in the future. And also we would like to invite patients and families and BCRT supporters back to our labs for another visit when we can. We don't know when this will be. I just want to get back in my lab at the moment, but once we're nice and settled and back in the lab, we'd love to have you all back and show you around. So yes, thank you. Yes, so have we got some questions, Zoe?
So I hope you'll agree with me that that's some really, really exciting results that Fiona and Tyler have shared with us. It's really, really promising work in Ewing-sarcoma. At this point, I'd just like to mention Ewing-sarcoma Research Trust. So we have partnered in funding Ewing's research for a number of years now, and the amount of work we do in Ewing's wouldn't be possible without their help and support and partnership. So a big shout out to Nigel. I think he's on the webinar. Thanks very much for all your support. So I'll hand over to my colleague Vicky, who's been collating the questions, and she'll put your questions to the experts.
Hi. So we have received some questions in advance for you. So I'm going to go through those first and then move on to the ones that people have been asking throughout the presentation, if that's okay. So the first one is that we've got a question from Vicky Woodall. And she's asking, are children eligible for the type of Ewing's immunotherapy that you've discussed this afternoon?
Okay, so should I take that one, Tyler? At the moment, oncolytic viruses, obviously they're undergoing phase, one of them is accepted in phase 3 trials, many of the others are still in phase 1 safety studies. Usually children were exempt from a lot of those studies to start off with for fear of how they were going to react. So actually, but more recently, some children are being treated with oncolytic viruses, but it tends to be in the context of neuroblastoma and the brain cancers, where a lot more of this preclinical data and research is out there in the forefront and for everybody to look at. So in the context of Ewing's, patients aren't being treated with oncolytic virus therapy, which is why Tyler's working hard in the lab to get that data. But it is progressing that children are starting to get these therapy in early phase clinical trials. What we would hope is that actually by the time we've got the preclinical data, there'd be more safety evidence in that cohort of patients, so we could progress things a little bit quicker. But at the moment, Ewing's patients aren't being treated. Hopefully that answers that question.
And there was a follow-up question from Vicky Woodall that could this type of therapy be considered something that could be used as a post-treatment that will help prevent children or in general patients for relapsing for Ewingssarcoma?
Yeah, I mean that would certainly be how we would anticipate the therapy, I think, at this stage, because of the immune surveillance and the importance of that in metastatic disease. I think we would anticipate that it would be used more as a therapy to prevent relapse disease and target metastatic or minimal residual disease that remains after primary treatment. I think that's how we would see it. I'm looking for Graham to start nodding at me. Yeah.
So we have another question which also was sent in advance, and it's a slightly related to a clinical trial, but I thought I'd mention it anyway. So there is a trial called Vigil, which it's a combination of a cancer vaccine with a couple of chemotherapy drugs, which are temozolomide and irinotecan. And so could you, do you know if this is sort of so the question refers to whether you could comment on the progress of that trial.
So I've read the papers about the Vigil immunotherapy trials, and they've provided some evidence of an anti-tumor immune responses, which is good. But as with any clinical trial, obviously it's still somewhat of a question mark, and that will probably be a question better suited to be answered by a clinician. Obviously, any clinical trial-based questions, we're all researchers. So if you want to know, I can tell you how Vigil works. For example, so it's taking tumour cells from the patient, and you basically edit them so that they express immune stimulatory molecules, and it inhibits enzymes which inhibit the production of tumour promoting molecules, basically. So we know how they work, but the clinical details we're probably not the best people to ask about, if that's okay.
Yeah, so I just wanted to follow up. So if so we can, for example, collect some information about clinical trial and send and follow up the question with to some of the people who are interested. So we're happy to do that if it helps. There's another question which it's from Gunvor Nygaard. So I apologize if I pronounce it incorrectly. It's, do you know if people are working on using nanoparticles to treat Ewingssarcoma?
Have you come across that?
I have, yes. So there are some preclinical papers, and anyone please correct me if there are clinical papers, but I've only come across preclinical lab-based papers, and I believe it's nanoparticles targeted against CD99, a molecule which is highly expressed on Ewingssarcoma. Personally, that's the only paper I've come across, but again, it's similar to what we're doing. It's preclinical work in the lab. I don't know. I couldn't answer about any clinical trials based on that.
Can I just comment, Talia? You're absolutely right. Good literature reading you've got there.
Lockdown.
So those studies are all preclinical stuff. Katya Scotlandy from Italy is really driving those sort of nanoparticle CD99 targeted therapeutics. And the strategy and the idea behind targeting CD99 is that there's some evidence that CD99 is involved in the metastatic process. So this might be similar to, you know, the sort of strategy you're developing, targeting the metastatic disease that's responsible for progression and relapse. Good background reading though, Tyler, that's great.
I was just looking at Tyler because I know she's been doing a lot of reading.
Brilliant. So move on to questions that people have asked this afternoon. So the first one is from Nicholas Kalita. So he has 2 questions. So the first one is, what are the surface proteins targeted on the tumor cell surfaces?
Is that with respect
For the viruses or the
Yeah.
proteins for antibodies?
I'm not sure. Maybe Nicholas can clarify.
Nicholas?
This is picked up some big words.
Cancer surface proteins.
Cancer surface proteins.
Oh, I'm interested.
Hi, I meant one of the proteins on the surfaces of the tumour cells.
Okay. So in terms
targeted?
sorry, Tyler. So in terms of how the viruses recognise the cancer cells for entry, the different viruses use different receptors, which might be why you get different efficacy in different cancer cell lines. So reovirus uses functional adhesion molecule, which is ubiquitously expressed. HSV uses a range of different receptor molecules, so HBM and Nectin-1, and the rhabdovirus uses LDR.
It's called low density lipoprotein receptor.
Yeah. So, but in terms of the surface proteins that are targeted by T cells,
Yes, to me, it's just science talk. It's not really useful. Some of this is a bit.
Yeah, not really, just one.
Yeah.
So so there was a second question from Nicholas, which refers to what are the differences in the way the DNA and RNA viruses operate in these experiments?
Okay. Do you want to answer that, Tyler? Or?
Yeah, I can do. So all of the viruses that we examined apart from the HSV are RNA viruses, and HSV is the herpes virus is a DNA virus. They are all molecularly distinct, but the main thing that determines how they all act differently in these experiments is, like Fiona just mentioned, the entry receptors which are associated with them. So what I've also done is, on each of these cell lines, I've checked the expression of these proteins called entry receptors on the surface of the Ewingssarcoma cells. So if the cells have high expression of these proteins, it's more likely that the virus will enter that cell. With the enterovirus, for which the cells were resistant, the cells don't express that entry receptor, so it's unsurprising that the virus can't enter the cell. Does that make sense? So yeah, it's not really the molecular detail, it's more the entry receptor which determines how they all act differently on the Ewingssarcoma cells.
And can I just follow up to that as well, Tyler? I think the difference on how they enter the cancer cell is one thing, but what we also think about in the lab is how those viruses engage with the immune system. And what we do know is some of the viruses act via different immune mechanisms. So the Coxsackie virus or the enterovirus that Tyler had mentioned, that actually utilizes plasmacytoid dendritic cells to activate the immune system, whereas something like reovirus might use monocytes. So actually understanding the tumour microenvironment as well and what cells are available for the viruses to target is also a really important element of what we would like to do.
So there is another question which I think you've already answered, but I'll just ask again. So this question from Angela Batikis that says, what is so different between these viruses that rhabdovirus seems so much more effective than the others?
Yeah. So again, it's back to the entry receptors, like the viral entry. If the virus can't enter the cell, then in terms of the direct killing at least, because for the immune aspect that's not the case, but for the direct killing, if the virus can't enter the cell, it's unable to lyse the cell. One really important aspect about the rhabdovirus is it has a replication cycle of around 6 hours, so it's able to enter one cell, and this is why we see such high effects at low doses. So it enters one cancer cell and is able to replicate and make multiple progeny virus particles within 6 hours. Whereas the other viruses have much longer virus replication cycles. So yeah, I would say that's the thing that makes the rhabdovirus more efficacious in terms of the Ewingssarcoma. Yeah.
There's another question which I get also refers to why did you choose this virus? So it's a question from Matt. So he's asking, I don't know if you want to ask yourself, but he's asking why did you pick these three viruses, these viruses with the start? Why did you pick those?
So these viruses have all, I think Fiona did touch on this, but so HSV, not the particular, a HSV virus has been approved. So obviously you want viruses that are clinically advanced. That was the main, in the grant that was written for this project. So basically you want viruses which are more clinically advanced so that if we do find promising results, then they can take them forward into clinical trials more quickly if they're already found to be safe. So all of the viruses that we have selected have all been tested in clinical trials. The enterovirus up to phase 2, reovirus has been up to phase 3 clinical trials in head and neck cancer, and a HSV virus has been approved. The rhabdovirus is in a phase 1 clinical trial. So I think that is the main reason. There are other viruses which are being clinically investigated, but we looked at these ones first and we found some promising candidate viruses, so we focused on those.
There's one further question from Nicholas Calista, and he's asking, do any of these viruses operate by directly modifying CD8+ genomes to code for novel HLA-1 molecules?
Okay, well, I'll take that one, Tyler, but I suppose the short answer is we don't know. I think we know that the viruses don't replicate or infect the T cells directly. The only immune cells that the virus seems to really enter are the more phagocytic cells, so the monocytes, dendritic cells, where we see low level infection. So my guess is directly they probably wouldn't do that. But what we do know is when they're engaging with the antigen presenting cells, that you can prime antigen specific T cells. And we've done that in quite a few other different tumour models now. But whether that switches kind of the T cell receptors and other at more of a genomic level, other than looking at what they recognise, that's probably as far as we've got with those studies.
And we have one final question, which is more a comment than a question really, is whether you will venture to put a timeline into when would these therapies have a direct application to patients?
Oh
yeah,
so that's a hard one. Obviously the viruses we've got are to clinical grade. They're owned by by biotech companies at the moment, so I think it's a case of kind of getting the preliminary data that's on board and getting the companies engaged in the promising data that we have to make sure that they can get put forward to clinical trials. I think I think if the data's there, the capacity's there, they can bulk the viruses up to clinical grade, but I don't think we'd like to put a time on it. Ideally, if I was guessing, it's going to be years rather than months. So that's a very vague answer.
I think that covered all the questions so far.
I've just seen another question that was a really good question. Am I okay to?
Is that about immune medicine?
Yeah. Someone asked about is this work in vitro only or in vivo? It's a very good question. Obviously animal models provide really great representations of disease. Unfortunately there is no animal model for Ewingssarcoma, as the genetic cause, which is the chromosomal translocation, is embryonic lethal in mice, which means that you can't generate the mouse models unless they're immunocompromised, in which case they're no use for investigating immunotherapy. So I just thought I'd touch on that. Obviously, that kind of work is really important, but it's not quite possible in immunotherapy investigations for Ewingssarcoma.
Yeah, and I've just seen another question from Nicholas about stimulating immune memory. Now, we know in animal models they do stimulate immune memory, so we can do re-challenge experiments in melanoma, and you have kind of those recall responses, which is excellent. Obviously, in the context of Ewing's, that's difficult because, as Tyler said, there isn't the in vivo models to be able to do that. But what we can do is phenotype the T cells that we generate in the lab and start looking at some of those memory markers, which is something that's on the to-do list but we haven't done yet.
Fabulous. I think, have we got through all the questions now, I think? So I'd like to thank our two speakers, Tyler and Fiona, and also to Graham Cook, who is Tyler's other supervisor, who's been on the webinar. So thank you for joining us and sharing your updates of your work. It's really, really good to see the progress that's being made. Next week, we have another webinar, and we have another of our PhD students, Hannah Spencer, who will be giving an update on her project, which is looking to adapt methotrexate in osteosarcoma, and she will be joined by her supervisor, Professor Rob Faulkner. So thank you to everyone for joining us, and we hope to see you again next week. Bye.
Thank you. Bye.